Deep within the caves of Yunnan province, China, a silent threat lurks. Chinese researchers have discovered a new bat virus named R.P.1.06. This discovery is a stark reminder that the microbial world holds many secrets, some of which could pose significant threats to human health. R.P.1.06 is a coronavirus, the same family as the virus responsible for the COVID-19 pandemic, SARS-CoV-2. While not an immediate cause for alarm, the discovery of R.P.1.06 underscores the need for continued vigilance in monitoring viruses emerging from wildlife. Bats, in particular, are known reservoirs for a wide variety of viruses. Their ability to fly long distances and their social behavior make them efficient vectors for viral transmission. The discovery of R.P.1.06 in bats is a reminder of the interconnectedness of human and animal health. What affects one can easily affect the other. The COVID-19 pandemic demonstrated with chilling clarity the devastating consequences of a virus jumping from animals to humans. The emergence of R.P.1.06 serves as a potent reminder that the conditions that allowed for the COVID-19 pandemic are still present. We must remain vigilant and proactive in our efforts to detect and understand these viral threats before they escalate into global crises. The most concerning aspect of R.P.1.06 is its ability to use the same cellular entry pathway as SARS-CoV-2. Both viruses target the ACE2 receptor, a protein found on the surface of human cells, to gain entry. This shared entry mechanism is a cause for concern, as it suggests that R.P.1.06 may possess the potential to infect humans. The ACE2 receptor acts like a doorway into our cells, and both SARS-CoV-2 and R.P.1.06 have the right key to unlock it. This shared entry mechanism is a product of viral evolution, a constant arms race between viruses and the hosts they infect. Viruses are constantly evolving to exploit vulnerabilities in their hosts' defenses. The fact that R.P.1.06 can bind to the human ACE2 receptor is a red flag. It suggests that this bat virus has the potential to jump the species barrier and infect human cells. While this does not necessarily mean that R.P.1.06 will cause a pandemic, it highlights the need for further investigation. While R.P.1.06 shares the ACE2 entry pathway with SARS-CoV-2, it is genetically distinct. Initial analyses indicate that R.P.1.06 is not a direct progenitor of SARS-CoV-2, meaning it is not a close relative. However, the genetic similarities and shared entry mechanism warrant further investigation. Scientists need to determine the full genomic sequence of R.P.1.06 and compare it to other known coronaviruses. Understanding the genetic makeup of R.P.1.06 will provide valuable insights into its origins, its evolutionary relationships to other viruses, and its potential to infect human cells. This information is crucial for assessing the risk it poses to human health. Scientists will also need to study the behavior of R.P.1.06 in laboratory settings. Further research is needed to determine if R.P.1.06 can replicate efficiently in human cells and if it can evade the human immune system. These studies will be crucial in assessing the potential threat of R.P.1.06 and in guiding the development of vaccines or antiviral treatments should they become necessary. The more we learn about R.P.1.06, the better equipped we will be to respond to the threat it may pose. The discovery of R.P.1.06 is a stark reminder that the next pandemic could be lurking just around the corner. We must remain vigilant in our efforts to detect and understand emerging infectious diseases. This requires ongoing surveillance of wildlife, particularly bats, for novel viruses. It also necessitates enhanced laboratory capacity to rapidly identify and characterize these viruses. The world needs to invest in pandemic preparedness. This includes developing a global early warning system for infectious diseases, strengthening public health infrastructure, and supporting research into vaccines and antiviral treatments. We must learn from the lessons of the COVID-19 pandemic. A coordinated global effort is essential to prevent future pandemics. International collaboration, open sharing of scientific data, and investments in public health infrastructure are crucial. The discovery of R.P.1.06 is not a cause for panic, but a call to action. We must remain vigilant, proactive, and united in our efforts to stay ahead of the next pandemic.